A number of previous studies have demonstrated a causal relationship between cannabis exposure and changes to brain morphology. However, most of this research is accomplished using adult participants who are heavy users of cannabis, even though the majority of people's interaction is recreational. This is why the main focus of this study will be determining whether gray matter volume predicted behavioral features previously associated with cannabis use and with the ECB system is shown in adolescents with just 1 to 2 instances of cannabis use. The endogenous cannabinoid system helps facilitate maturation-related neural reorganization and is a way that cannabis can produce neurobiological changes. This system is a key factor in the study's hypothesis. Our hypothesis states that we predict that even extremely low levels of cannabis use would be associated with structural brain differences in regions previously implicated in cannabis use studies and in the ECB system, which consists of the amygdala, hippocampus, striatum, and the cerebellum. To complete this experiment, 2 cohorts of participants are needed. Cohort 1 consists of 46 participants who only had 1 to 2 lifetime instances with cannabis at baseline testing. In order to test whether observed differences between cannabis users and controls may precede cannabis use, Cohort 2 was created. This cohort consists of 69 participants who were cannabis naive at baseline, but reported using cannabis at least 10 times 2 years later at follow-up testing. Whole brain voxel-based morphometry approach was used as it allows more extensive testing of regions in the ECB system, such as the frontal cortex and the posterior cingulate. The gray matter volume of both cohorts was tested and examined. We then compared the spatial distribution of the GMV effects using biomarkers of the ECB system. The first is the CB1 receptor availability, taken from a previously published independent sample, and the second is the expression of the CNR1 gene, which encodes this receptor, taken from the Allen Human Brain Atlas. The results from this experiment were consistent and helped to prove the hypothesis to be correct. Through the voxel-based morphometry testing, it was found that the gray matter volume of Cohort 1, participants who had 1 to 2 previous interactions with cannabis, was significantly greater than those of their control counterparts. This difference was seen in several different brain regions, including large medial temporal clusters such as the amygdala, hippocampus, and the striatum. It was also seen in the lingual gyri, posterior cingulate, and the cerebellum. As the ECB system is composed of the amygdala, hippocampus, striatum, and the cerebellum, all structures where the gray matter volume increase was seen, one can draw the conclusion that the spatial distribution of this increase can be associated with the endogenous cannabinoid system, which then indicates that cannabis exposure may be the cause of these findings. In this figure, line A demonstrates the brain structures of Cohort 1 that have increased gray matter volume. Line B are outlines of the anatomical regions superimposed on a binarized mask of voxels showing Cohort 1's greater gray matter volume. In line A and B, the brain slices are in 15 millimeter increments, and from left to right, they start at the most anterior slice and go to the most posterior slice. In line C, the brain slices are taken in 6 millimeter increments and are organized from left to right, going from the most inferior slice to the most superior slice. These slices demonstrate outlines of striatal subregions superimposed on a binarized mask of the voxels, showing Cohort 1's increased gray matter volume. This figure further demonstrates the increase in gray matter volume of those who had 1 to 2 previous encounters with cannabis compared to their control group. Regarding the results of Cohort 2, the participants who were cannabis naive at baseline but had at least 10 interactions with cannabis at follow-up testing 2 years later, there were no gray matter volume differences when comparing them to their control group at baseline testing. Overall, the data collected from this experiment is extremely beneficial and significant in helping prove the causation of cannabis usage and the effects it has on gray matter volume in several structures of the brain. This study has become increasingly relevant and important as of late. In the past, most studies that have been done regarding the topic of cannabis focus on adults who have a long-term history of heavy cannabis usage. With the recent changes in the legal status of marijuana, as well as the increasingly relaxed view of the substance, the rates of adolescents using cannabis have been steadily increasing. About 35% of American 10th graders have used cannabis, and with the more research being done about the negative effects of it, this study was important in furthering the knowledge of the effects of cannabis on the maturing adolescent brain. The results of this study draw similar conclusions to several studies that have been previously completed. One in particular is the Avon longitudinal study of parents and children. This study demonstrated results of increased cortical thickness in adolescent males that had less than 5 interactions with cannabis compared to their control group who never had any instances with THC. With the converging evidence, gray matter volume differences were not caused by group differences such as demographic, personality, psychopathology, or any other substance use factors. In our study, an examination of Cohort 2 did not display any gray matter volume differences, which suggests that the gray matter volume differences do not predate cannabis usage or any other unidentifiable factors. The implications of this study are the opposite of what is currently being promoted more recently about the substance. As of late, cannabis has been seen in a better light, being non-addictive and having helpful properties for some. Though this may be true, it is still important to note that there are still negative effects to the substance. This study has demonstrated how the brain is negatively affected, increasing the gray matter volume in several brain structures, but also shedding light on its increased effects on an adolescent brain, only after a few instances of using the substance. Using voxel-based morphometry, the gray matter volume was increased in extensive regions of the brain. The analysis of the collected data helped to prove the gray matter volume differences between Cohort 1 and Cohort 2 against their respective control groups. It was also proven that it was unlikely caused from a factor that predated the cannabis interactions. The study was completed very professionally with an immense amount of controls and regulations to ensure the data was pure. If there were to be any potential changes to the study, they would be minor. Participating subjects chosen were racially and ethnically homogeneous. So one change or expansion of the experiment would be to potentially choose participants with a more diverse background. Another potential change would be adding additional ways to measure brain structures for further confirmation of the data being collected. Future studies associated with this topic would be to separate the results by gender and compare results this way, or assess other ages and their gray matter volume after cannabis interaction. Overall, this study is extremely relevant and important to society and its current state. Increasing awareness of cannabis and its negative effects on the adolescent brain need to be publicized at a higher rate so that more people can understand the truth about what this substance is.